So, Assalamu alaikum and a very good morning. I, Dr. Amnaskar, welcome you all to the invited lecture session of First International Conference, Trends and Research in Chemistry, organized by Department of Chemistry, Division of Science and Technology, University of Education, Lahore. Dr. Munaza Shahid will be co-chairing this session with me. So this session is actually already started at 11 and will finish at around 1 p.m. There will be 4 talks today in this session, and every speaker will have almost 30 minutes for their talks and discussion. First talk today in this session will be delivered by Professor Dr. Adeel Afzal. Professor Dr. Afzal has studied chemistry at University of Vienna, Austria, and earned his PhD from there in 2007. He did his postdoc from University of Bari, Italy. In start of his career, actually, he served as assistant professor at Qasim University, Islamabad, and Comsets University, Islamabad, in the Lahore campus. And then he moved to Saudi Arabia, where he served at King Fahd University of Petroleum and Mineral and University of Hafar al-Batin for last 9 years. Currently, he is professor of journal and inorganic chemistry at the University of Punjab, Lahore. In 2021, Professor Afzal was admitted as a fellow of Royal Chemistry. Is there anything wrong with the voice? Okay. He is also ranked as the world top 2% highly cited scientist for the year 2020 with a composite citation score of 3.06. His research interests include the development of imprinted and functional materials for chemical and biosensors, biomedical diagnostics, environmental and energy-related applications. Dr. Saab, please, over to you.
Thank you very much for a very nice introduction, and thank you very much for giving me this opportunity. I will just switch off my cam to so that it doesn't bother the network. Okay. I hope you can see the slides.
Yes, thank you.
Okay, so I'm going to present some of the examples of our research work we have already performed in our labs on the development of synthetic antibodies for environmental process control and diagnostic applications. So basically, we develop devices, sensors for the detection of different analytes depending upon the application. So first of all, the contents of this presentation, they are divided into 3 parts. So I will briefly introduce you to the topics like what are natural antibodies and sister phenomena, what is molecular recognition, then what are synthetic antibodies and what are our strategies, how can we develop these synthetic antibodies. Then, of course, when we develop the devices, so we need the transducer that can translate the chemical information into electrical signal. So we have used different types of transducers. I'll just go through these few examples. And then our main discussion will be on the applications. So I will discuss environment, in which we'll discuss air quality monitoring. Then we'll go to process control, how we can control different processes, and disease diagnostics in the end. First of all, what are natural antibodies and how do they work? So, thanks to this pandemic, so I hope most of the people are familiar with antibodies now after COVID. So natural antibodies are basically the first line of defense in our bodies or in organisms. Now, they are responsible for innate and adaptive immunity. So innate antibodies are present in our bodies since birth, so they are a part of our genetic makeup, you can say. So and they are broad spectrum antibodies. They are usually non-specific, and they can counter different pathogens. Now, adaptive antibodies are more relevant here because adaptive antibodies, they are very specific to a certain kind of antigen. For example, the vaccination we got for COVID, it develops the adaptive antibodies in our system that can particularly interact with that coronavirus. So adaptive antibodies, they are very specific, and here you see, because antibodies are proteins, immunoglobulins, so this is usually they are depicted like this. So here you see an antigen that is selectively captured at this point here by the antibodies, and this is how these antibodies work. Now, when we take this phenomenon in chemistry, we study molecules and their interaction. So in chemistry, when you have an analyte, like here in this figure, you may see that this is isophthalic acid, and isophthalic acid has carboxylic acid groups. So C double bond OOH groups. So here we have oxygen and hydrogen. So how these molecules can interact with receptors or other molecules, this phenomena is called molecular recognition. And usually molecular recognition is based on non-covalent interactions. So non-covalent interactions between two molecules. So these interactions can be electrostatic, can be hydrogen bonds, can be van der Waals forces or other kind of forces. So in this figure, basically you see a receptor molecule that interacts with isophthalic acid through hydrogen bonds. So the receptor molecule here has the heteroatoms, which can interact with the hydrogen of the carboxylic acid, or here we have hydrogen that can interact with the oxygen of the carboxylic acid in isophthalic acid. Now, how do we translate these two phenomena, or how do we take these two phenomena into our research is to prepare how we prepare these antibodies. Synthetic antibodies, these are artificial receptors, and you can say these are mimics of natural antibodies that can interact selectively with any analyte, and then they can recognize it. Here in this figure, you will see you're seeing a pyrene molecule, a polycyclic aromatic hydrocarbon here in blue color. So this is a pyrene molecule, and it is surrounded by the receptor molecule. Mostly in yellow color, you see the receptor molecule. So this receptor molecule develops pi-pi interactions with the pyrene, and it envelops this pyrene molecule inside. So I will show you how we have prepared this one. So these interactions, just remember that these interactions should be non-covalent, and these antibodies, even if we are preparing them artificially, they should have certain specificity. So if we are preparing an antibody for pyrene, so for the detection of pyrene, it should mostly detect pyrene, and it should not be responsive to all other types of the molecules. So this specificity is very important when we discuss certain applications or when we address certain issues. How do we prepare these synthetic antibodies? Synthetic antibodies, basically, they are polymers can be based on nanomaterials as well, or ceramic nanoparticles. So we use a technique that is called molecular imprinting. Molecular imprinting basically makes use of a template molecule. A template molecule could be the analyte of interest, or it can be a similar molecule with a similar geometry. And then this template molecule, for example, in this case, we have pyrene here. So this pyrene is our template, and we have developed a polyurethane. So polyurethane, we have the monomers here, bisphenol A and fluoroglucinol, the cross-linker, as well as we have diisocyanate diphenylmethane. The template should be a molecule that does interact or that does not participate in the polymerization. So it should not covalently bind with the polymer chains. So there should be only non-covalent interactions. So we assemble the template, we put the template inside the monomer solutions, and then we polymerize it after some time. Then it's a process like a self-organization process or self-assembly process, in which the pyrene is surrounded by the monomer molecules, and then they are converted into polymer chains. So here you see the pyrene that is surrounded by the polymer chains. Because the interactions are essentially non-covalent, that means we can easily remove these pyrene molecules from the polymer. When we remove these pyrene molecules from the polymer, we get a cavity. So this cavity is called imprint, or you can say molecular imprint. This imprint is very specific. Just like natural antibodies, it is very specific to the pyrene molecule. So any other molecule, if it comes there, it will not have the same kind of interactions as pyrene, because this was a geometrical fit for the pyrene. Now we take this molecular imprinting phenomena and the synthetic receptors or synthetic antibodies, and then we apply these synthetic receptors on different devices or transducers. So here you see the piezoelectric or mass-sensitive transducers based on quartz crystal microbalance. We have used monoelectrode, dielectrode, trielectrode, as well as tetraelectrode quartz crystal microbalances. Quartz crystal microbalances, when you coat them, these are the gold electrodes on each of them. So you see here, this is the monoelectrode system, this is two-electrode system. So when they are coated and when they interact with any analyte, just like pyrene, so what happens, the mass increases on the surface, and the increase in mass is translated as a decrease in frequency. So the sensor will show you a decrease in frequency that will be related to the amount of analyte. And on the other side, we have electronic transducers made of interdigitated capacitors. Again, these are coated with these synthetic antibodies, and we can use them for the sensing. In this case, we will measure the capacitance. Now, the applications, the first application related to the environment is an example that we have already performed, and it was the detection of formaldehyde and volatile organic compounds. So I will quickly go to the slide where we have, this is basically the setup, in which we have a transducer here, the sensor device in a sensing chamber, and here the gases are mixed with the organic vapors. So air is mixed, or nitrogen is mixed with the organic vapors, and these organic vapors, they are exposed to the QCM device, and QCM device then detects the organic vapors, and we see a decrease in frequency through this oscillator circuit, and we record this data in the computer. So basically, I will show you how we have designed our synthetic receptor for formaldehyde detection. For formaldehyde detection, we have used paraformaldehyde as the template. So formaldehyde is produced from paraformaldehyde burning. So then we put it in ethylene glycol dimethacrylate and methacrylic acid. When it develops certain interactions inside the monomers, then monomer solution, then we polymerize these monomers and we get a molecularly imprinted polymer. So in this, we have also included gold nanoparticles, and gold nanoparticles were prepared separately, but they were fabricated on the crystal microvalence like this. So first of all, we have a gold electrode. On the gold electrode, we have coated the molecularly imprinted polymer layer. After that, we have coated a gold nanoparticle layer, and then we have coated a molecularly imprinted polymer layer again. So this is a layer assembly of the receptor layer or synthetic antibodies. And here you see the damping spectra of the device, which shows you also the thickness of the layer. Now, the results of these synthetic antibodies, how the antibodies interact with different volatile organic compounds. So here we have formaldehyde. You see for formaldehyde, the antibodies show the highest response, and it is, I think, double than the response of acetone. So they are very selective toward formaldehyde. If we test other volatile organic compounds like methanol or humidity, water, so these antibodies do not respond. And you see here a very important parameter, that what is the response time and recovery time. So these response times and recovery times, they are very good. So these devices are very efficient. So they can detect formaldehyde in less than a minute or less than 30 seconds. And then you can recover those devices in less than 13 seconds or around 13 to 15 seconds. So because we have the non-covalent interactions, that's why we can remove the formaldehyde that is already bound in the device very easily. I will go to the next application, and that is process control. In process control, basically, we have it's a two-way strategy. So we have to monitor a process, and then we have to adjust a process. So monitoring is done by the sensor, and adjusting should be done by some other means. So we have prepared, I will quickly show you this, that we have prepared the sensor, but the sensor here, now this process that we have done is from the oil refining. And oil refining, why we have chosen this process, we wanted to reduce SOx emissions, sulfur oxides emissions. And sulfur oxides, basically, when the fuel burns, sulfur oxides are produced by the sulfur-containing compounds that are present in the fuel. And most of these compounds are benzothiophene or dibenzothiophenes, and their derivatives like methyl substituted or alkyl substituted derivatives. So we have chosen benzothiophene and dibenzothiophenes as our analytes, and then we have developed these sensors for benzothiophene and dibenzothiopene. So basically, this sensor was based on titanium, sol, and nickel sulfide particles. So we have a composite, and we have coated this composite on interdigitated capacitor. It's a very small device, and we have exposed it to the fuel to detect benzothiophene and dibenzothiopene. And second strategy was to prepare membranes, and these are the membranes, molecularly imprinted polymethyl methacrylate and nickel sulfide composite membranes. So you can see here the real-time picture of this composite membrane. So again, these membranes were soaked into the fuels, and the quantity of or uptake of the dibenzothiophene was measured. So here, this is the result of the device. So the device shows excellent sensitivity, and it has a very low detection limit, 12 to 16 ppm for dibenzothiophene and benzothiophene. Then we have recovered around 73.5% of benzothiophene from the spiked samples of spiked fuel samples, spiked gasoline samples. And dibenzothiophene recovery was around 97.7%. For the membranes, here, how we can adjust the process, how we can reduce the benzothiophene or dibenzothiophenes. So we have measured the UV visible spectrum of the fuels. So these are the spiked fuel samples, and this is the lambda max for dibenzothiopene. Up to 1 ppm by weight solution, we were able to recover around 96.6% and around 97% of dibenzothiopene. So these values, they are corresponding to each other, and 96.6 or 97.7, this is approximately 97%. And we have determined these values from the absorbance intensity or the area under this curve. So we have used both methods and just to validate the results. And last is how we develop devices, synthetic antibodies for disease diagnostics. And we have chosen alpha-1 acid glycoprotein for this purpose. Alpha-1 acid glycoprotein is a biomarker for the early diagnosis of the cardiovascular diseases or diabetes, and it's sometimes also related to the hepatocellular carcinoma. So basically, if there is the presence of cardiovascular disease, the concentration of alpha-1 AGP, this glycoprotein in serum, will increase. Then, in this case, we have developed graphene oxide, reduced graphene oxide, copper oxide particles. And these particles were mixed with the ethylene glycol dimethacrylate, and we have used a functional monomer here with boronic acid as the functional moiety. And we have also used this alpha-1 AGE as the template. As a result, we have fabricated the layers on the QCM. So here we have the QCM and the layers. When you remove AGB, of course, you will get the cavities that will be particularly sensitive to the alpha-1 glycoprotein. So here are the results of our study. This is a very recent study, and we have just published in the last year. So we were able to detect it, and we have shown a series linear detection range that is around 1 to 200 nanograms per liter. This is the result of the detection in the solution. So here, then the detection limit that was shown was 0.25 nanograms per ml. But in serum or the spiked serum samples, we also tested this device. And here you can see that in serum, when we spiked the serum samples with 150 or 200 nanograms per ml of the alpha-1 AGP, we were able to recover or to detect around 90.71% and 92.98% of this. So this device works in serum also, and it works because serum is not like our solution. It's a very complex mixture, and it contains many other analytes. which can have non-specific interactions with the polymers. So here we have tested some of these interfering allies like bovine serum albumin, a protein, then a glucose or fructose or lysozymes and uric acid, and you see that this device was, I think, very much responsive to the alpha-1 AGP, so it gives a selective response towards this alpha-1 AGP, the acid-like protein. Now, I think Professor Franz Richard from University of Vienna, because he provided us the setup for the testing of the volatile organic compounds, and Dr. Peter Lieberzeit, and Dr. Adnan Mujahid, my collaborator at EU, Dr. Nasir Iqbal from University of Afzal, and Dr. Usman Latif at the Comsat University of Lahore, also to my students, and to the fund from Ministry of Education of Kingdom of Saudi Arabia and University of Afzal, Deanship of Scientific Research, as well as to the Higher Education Commission and University of the Punjab for funding and providing us facilities. So thank you very much. If you have any questions, please.
Dr. Saab, thank you very much. A few questions, please. First question is, in the talk, you said that these are mimic antibodies. Actually, these synthetic antibodies actually 100% behave like the original natural one, or there is some difference, or at some points there is any difference between the behavior of the two?
Actually, it is not possible to mimic the nature 100%. So of course it is not possible. So what we do, or what this molecular imprinting does, is that it brings sufficient selectivity to the polymer. So if we take just polyurethane and expose it to any other analyte, so of course there can be, there will be non-specific interactions of the urethane group with other groups of the analyte. When we imprint pyrene or any other analyte into that, that gives us extra selectivity and extra sensitivity towards that specific analyte. How? Because it contains the cavities that are particularly geometrically and functionally fit for those analytes only, those candidate molecules only. So, of course, there are always non-specific interactions, as I have shown in the last slide. Some of the BSAs are also detected, some of the lysozymes are also detected, but these are non-specific interactions, and non-specific interactions will always be there.